By the time you watch this video, you should have a decent understanding of all the steps involved in stimulating a muscle fiber to contract. If you don't, pause, go back, and take a look at that first. In this video, we're going to talk about what happens when a muscle relaxes. So what happens when that muscle is no longer getting the signal to contract. We are all familiar with the experience of a muscle relaxing. You know, we pick up that heavy jug of milk, we set it on the counter, and our arm falls back to our side. But what's actually going on in your muscle at that point? Now, there are two things to consider in terms of muscle relaxation, two things that you need to understand. The first is stopping the signal. So when your muscles are no longer getting the message to contract, yes, your neurons stop firing, but how does that signal then get translated over to the muscle? You know, we talked about how the signal to contract gets translated over to the muscle with excitation-contraction coupling. So what happens after the neuron is no longer firing? What happens at the muscle fiber? The second part of muscle relaxation is the return to resting length. You know, after you set that milk jug on the counter, your muscles don't stay in the position that they were when you released the milk jug. Your muscles relax back to their resting length, which is longer than the contracted length of the muscle. So we'll talk about each of these. There are two things that have to happen for a muscle to realize, I'm using that in quotation marks, for a muscle to realize that it's not getting that signal to contract anymore. The first thing that needs to happen is the breakdown of acetylcholine. Remember that acetylcholine is the signaling molecule that's released at the neuromuscular junction, and that kind of takes that excitation from the neuron and communicates that over across the neuromuscular junction to the muscle fiber. It's released when the neuron is excited, when the motor neuron is excited, and it opens up, it binds its receptor and opens up a channel in the sarcolemma of the muscle fiber. In order for the muscle fiber to stop getting the signal, the acetylcholine has to go away. And there are actually a couple of different ways that acetylcholine is removed. It's removed by diffusion. Some amount of the acetylcholine just diffuses away from the neuromuscular junction. And it's also removed through enzymatic breakdown. And the enzyme that breaks down acetylcholine is acetylcholinesterase. Acetylcholinesterase is always present at the neuromuscular junction. It's always breaking down acetylcholine. It's just that when the neuron is active, there's more acetylcholine being released. So the amount of acetylcholine being released overrides the activity of acetylcholinesterase. But once the neuron is no longer firing, the neuron's not releasing any more acetylcholine. So now acetylcholinesterase wins, and acetylcholinesterase breaks down the acetylcholine that's left. Acetylcholinesterase is a really fast enzyme. It breaks down acetylcholine quite rapidly. So as soon as your brain stops telling your motor neuron to stimulate a muscle fiber, acetylcholinesterase breaks down the remaining acetylcholine, and the muscle fiber is no longer receiving that signal. Once the acetylcholine is completely gone, the ion channels in the motor end plate close up. Okay, so now the sarcolemma is turned off. It's not getting that signal from acetylcholine anymore, but you still have all those calcium ions floating around in the sarcoplasm, binding to troponin and letting the myosin heads grab onto the actin thin filaments. There is a second thing that has to happen in order for the muscle fiber to be able to relax, in order for the muscle fiber itself to stop contracting. And that is calcium reuptake. You've got to get that calcium back out of the sarcoplasm. And that calcium goes back into the terminal cisternae. It goes back where it came from. There is a pump that actually takes calcium out of the cytoplasm and swings it back up into the terminal cisternae. So it's active transport. It does require ATP. This is another reason why rigor mortis happens, that stiffening of the muscles after death. If your body is dead, you're no longer breathing, your heart is no longer circulating oxygen around your body, your cells can't make ATP anymore. And if your cells can't make ATP, two things happen. First off, your myosin heads can't detach from the actin thin filaments, so your cross bridges stay intact. But the other thing that happens is that your muscle cells cannot take calcium back up into the terminal cisternae, so the calcium ions just stay out in the sarcoplasm. But that's only after you're dead. If you are alive and functioning and your cells are making plenty of ATP, calcium gets taken back up into the terminal cisternae. And as the calcium is taken back up into the terminal cisternae, the calcium concentration in the sarcoplasm drops. So, and as that calcium concentration drops, the calcium ions leave troponin. They're no longer interacting with the troponin molecules. Troponin shifts back its shape and rolls tropomyosin back over the thin filament active sites, covering them up so that the cross bridges can't form. These are the two factors that stop excitation-contraction coupling when a muscle is no longer being stimulated to contract. You get breakdown of acetylcholine and you get calcium reuptake out of the sarcoplasm into the terminal cisternae. Of course, we all know that when a muscle stops contracting, it doesn't just freeze in place and stay there. The muscle actually returns to its resting length. It lengthens again. So there are three factors that play into that happening. The first thing that I want to emphasize is that this is a passive process. It does not require ATP, at least not in the muscle fiber itself that is actually relaxing. It is entirely passive. And it happens due to three things. There are three things that really contribute to this. The first thing is the elastic recoil of muscles and tendons. So there are elastic fibers in your muscle fibers that return to resting length once they're not being contracted. There are also elastic fibers in your tendons, in the connective tissue that packages and around muscles and that connects muscles to bone. There are elastic fibers that also return to their resting length once the pressure is removed. Secondly, you have antagonist muscle contractions. So let's think about the biceps and the triceps as an antagonist muscle pair. When the biceps contract, they flex your arm. When the triceps contract, they extend your arm. So when you are, say, doing an arm curl with a dumbbell, you're using your biceps to bring it up. But when you stop contracting your biceps and you want to move the dumbbell back down, your triceps become active and they extend, and that extension actually pulls your biceps back to their resting length. Third factor is just gravity. You know, if you just relax every muscle in your arm and try your best to just get them as completely relaxed as possible, gravity is going to bring your arm down and return those muscles to their resting length. So those are the three major factors in actually returning your muscle back to where it started before the contraction.